Okay, welcome back everyone for we wait. Okay. Okay. Welcome back everyone for the second part of the dormancy session. And we have an announcement that after Jean-René's talk, after a couple of more talks, actually, like at 1 p.m., there's a lunch break for everyone, and it's going to be in exactly the same place where the coffee break was. So just please keep in mind. So, and our next talk on the dormancy session is Jean-René, and from Collège de France, and he's going to tell us about evolutionary adaptations. So.
Thank you. So first of all, I would like to thank Marie-Hélène and Elvan for this invitation here, and also the Fondation Polignac for organizing a meeting on reproduction and fertility, which really is a field that I find fascinating because of the diversity of mechanisms that one can find throughout the animal kingdoms. And this diversity can be found at every level, from behavioral level, as you can see here, down to the molecular level. Je pense qu'ici c'était encore. Ah non, c'est celui-là. Ouais, super. And in the lab, actually, we are trying to find what is if there is a logic behind this diversity, and what could be, the constraints that have shaped these different mechanisms to reproduce. J'en ai un là. Merci, super. And we focus on one specific stage of reproduction, which is meiosis, as shown here. And compared to mitosis, where you produce two daughter cells which have the same genetic materials between these two daughter cells and the mother cells, during meiosis, you have the production of 4 different cells with different genetic contents. And these contents are also different from the mother cells here. As shown here, the first step of meiosis is the two copies of each chromosome, one from the fathers and one from the mothers, need to move within the nucleus. They need to identify each other between all the other chromosomes. Then you have a double-strand break of the DNA, which can be repaired as a crossing over sometimes. And this actually leads to the formation of new gametes, the separation of the two homologs, and the action of chromosome information. You can see you have a bit of green here to the red chromosome, and then at the second meiotic division, you separate the two chromatids. And this led to this kind of scheme that you can find in the textbook, where you have leptotin, chromosomes start to condense, they move, as I said, formation of double-strand break, and pairing of the chromosome, formation of the synaptonormal complex, and then of the crossing overs. However, what I think is very interesting is that in the few organisms where it's being well described, the mechanism behind those different stages are quite different. And this starts really at the first step, when chromosomes move, actually. This is the example, for example, of Pombé, the fish and yeast, where you can see the nucleus moving back and forth to align its chromosome. And those movements are driven by the microtubule here, outside of the nucleus. And the nucleus is actually, the chromosome here actually tethers to the microtubule through the Sun and Cash protein at the telomere here. And in contrast, what has been shown, for example, in C. elegans, is that you don't, I don't know how to tack. How do I go back? No. Oh, this one, okay. And could you stop the movies? Is that possible? So in C. elegans, what have been shown by different labs is that you do not have global movement of the nucleus, but each chromosome actually moves independently. And here it was the example of the X chromosome, which is looking for its partner. And the movements are microtubule dependent, again. These chromosomes are linked through pairing centers to the microtubule through the Sun and Cash protein again, the Sun being at the inner membrane and the Cash protein being at the outer membrane here. And instead, what we've shown in Drosophila females a few years ago is that you have global movement of the nucleus. You have this rotation here, driven by microtubules, and in red what you have are the centromeres of each of these chromosomes. And we actually show that it's the entire chromosome which are moving, because we can label, for example, in red with a UV pulse, any part of the nucleus of the chromatin, and it will rotate. And as I said, those movements are driven by microtubule and also by the same machinery, the sun and cache domain. Here in green, you have a cache GFP protein rotating with the centromeres. So what it shows is that although the machinery seems quite similar between all the different organisms, you have the sun here, the cache domain, you have many differences. For example, as I said, it is driven by microtubule in most species, but it can also be driven by actin cytoskeleton in the budding yeast, for example. The link with the chromosome can be the telomere, the pairing centers, or the centromeres, as I said, in flies. And so to explore a bit further this diversity of how you move your chromosome, we decided to look in Drosophila to the Drosophila males, which may seem not so far apart from the females, but Drosophila males actually do meiosis very differently. I mean, they don't have SC, they do not have any double-strand break, but they still segregate their chromosome fine. So what Thomas did, the first thing he looked at was the movement of those chromosomes. And that would be nice if this movie plays. No. Yeah, okay. So too bad. This is, as a control, this is a female when you can see the rotation again of those chromosomes. And in the male, what he found that you have the same machinery, so centromeres are also clustered. You have the sun and cache domain in green colocalizing with the centromeres. But if you look at those movements, they are really small and they don't very slow. And if you quantify the volume explored by the centromeres in males, is actually very low compared to the females. It's about 3 or 4 times lower. And for us, it was quite puzzling because in the female, we had shown that in a sun mutant, where the movements are quite affected, the nucleus are still rotating, but the chromosomes are not linked to the nuclear envelope. In this case, you have lots of defects, you have lots of pairing defects and the defect information of synaptomal complex. Whereas the males is even lower and still manage to segregate its chromosome. So the solution to that puzzle actually came when Thomas measured the time that these males had to pair their chromosome, here in blue, and he found that males have about 100 hours to pair its chromosome, whereas in the female here in red, it's about 20-25 hours, so much less, 4 times less. So we had a nice correlation where females need to move their chromosome faster because they have a shorter period to pair their chromosome compared to males. But we wanted to have to demonstrate that this correlation was important by going back to the female. So we took a female which is a sun mutant in which the movement, as I said, are affected. And in this condition, we lengthen the cell cycle by downregulating cyclin B and CDK1, specifically during that pairing window. And we went up, so we could double the time. In the wild-type situation, it's about 20 hours. And if you downregulate cyclin B and CDK1, you are at 40 hours, or even 45 hours. And now, if you combine the two, the sun mutant and a delay in the cell cycle, we could actually rescue the pairing defect. So this is the numbers of unpaired chromosomes in a sun mutant. And you can see that if you downregulate CDK1 and cyclin B, you can rescue the numbers of chromosomes which are paired. So indeed, one conclusion of that part was that time seems to be quite a very important constraint to could explain the difference in mechanism exploited by these 2 different model systems, males and females. So females need to move their chromosome fast because they don't have so much time, and sperm have more time to pair their chromosome. And I think it could be also very interesting now to go back to all those different model organisms and to actually measure the time, the duration of the pairing window that they have. And that could be one of the explanations of why they have evolved so many different mechanisms to pair. And now I will move to the second step of meiosis. So after moving your chromosome, you induce the formation of double-strand break, which are used in many species to pair your chromosome and to form a synaptomal complex. The formation of the double-strand break, again, are made by a very conserved machinery based on SPO11, which is a topoisomerase which induces double-strand break. And these breaks sometimes can be repaired on the homolog chromosome by sequence complementarity, and this is used to identify the homolog chromosome for pairing. But not in all species. It's actually used, for example, in the mouse or in the zebrafish, also in the budding yeast. If you are, for example, in a SPO11 mutant, in this organism in purple, you are unable to pair your chromosome. However, it's not true in the one that I put in yellow. The Drosophila or C. elegans can pair their chromosome and form a synaptonormal complex without any double-strand break and in a SPO11 mutant. So what we decided to do to understand a bit further this difference of mechanism was to use a species at the base of these animals, because all those animals are actually, all those ones are bilaterian, so they have a left-right asymmetry. And we decided to work with one which is more basal, which is a jellyfish, which is a Nidarian called Clicia hemispherica. And in these ones, and this is the work of Catriona Munro in collaboration with Evelyn Olliston. And Clicia has a two-step in this life cycle. After fertilization, it gives rise to an embryo here, which can metamorphose on the seafloor as a polyp. And this polyp can reproduce asexually. You can cut it and it will just regrow. But it also produces some baby jellyfish here, which are the sexual form. And you have a females or males jellyfish, which is the swimming part, with the gonad here. And the nice thing is that we can actually induce mutation by injecting CRISPR-Cas9 and the guide RNA into the oocyte, and then generate homozygous mutant polyps that will produce homozygous mutant jellyfish. And so that's what we did for SPO11. And we look at the initial stages of meiosis, and you can see in the wild-type situation the nice bouquet of telomere at the leptotin-digotin stage here. And you can see that in a SPO11 mutant, you have the formation of the bouquet perfectly fine. All the telomeres are clustered on one side of the nucleus. However, then we look at pairing using probes that we made against the centromere part of the chromosome 5 and the telomere parts of the chromosome 5. And at the packeting stage, it may be a bit hard to see here, but you can see that the two centromeres and the two telomeres are perfectly paired in the wild-type situation. But in a SPO11 mutant, you can see that the two centromeres here are quite far apart, and the two telomeres as well, indicating that you cannot pair without SPO11. And also then we look at the formation of the synaptonormal complex, looking at the central part here, SYCP1, and the lateral elements, which are SYCP3, and you see that in the absence of SPO11, you form polycomplexes, but you cannot form a synaptonormal complex in the females, and it's the same in the males as well. So the conclusion was that Clicia is now part of the purple team, indicating that you do require SPO11 and double-strand break to make a SC and to pair your chromosome. Indicating that it could be the ancestral form of making meiosis in animals. However, I'm sure that some of you have already noticed that in the unicellular organism, you also already have that kind of difference. And Pombe does not require double-strand break to pair its chromosome compared to the budding yeast. And to us, it was also very puzzling to find that Clicia here is actually closer to, it's at the same time similar to budding yeast and to the vertebrate system. And so the best correlation we could come up with to explain this difference is when we look at the numbers of chromosomes. So when you look at this is the haploid type, so the numbers of pairs of chromosomes, you can see that Clicia has 15 pairs, which is quite close to the budding yeast, and to the mouse as well. Whereas the species which have less chromosomes, so such as Drosophila or the flatworm here, have a lower numbers. So we reason that these traits, whether you need double-strand break to pair or not, might not have a phylogenetic inheritance, but could appear if you instead of ranking these animals through the phylogenetic tree, if you rank them according to the numbers of chromosomes that they have, you can actually see that the one with low chromosome numbers do not require SPO11, and the one with a higher numbers of chromosomes do require SPO11. So it is a nice correlation, but how do you demonstrate that correlation? That's another problem. So what we decided to do was to look at many more species, and so it's a lifetime project. We will look at as all the animals that we could lend our hands. We will do a SPO11 mutants, but we had to start somewhere. So we decided to start with the nematodes because already some interesting work has been made, and they've shown that you already have some differences between C. elegans and Pacificus, for example, and also the nematode family has evolved quite rapidly. In terms of karyotypes and also in terms of reproduction modes. And we team up with the lab of Marie Delattre in Lyon, and we chose 4 different species of nematodes: Panagrolimus, which has 4 pairs of chromosomes, Oshayus 6, Mesorhabditis bellari 10, and Mesorhabditis longispiculosa with 17 pairs of chromosomes. And we are in the process of making SPO11 mutation in these different species. I don't have all the results yet, but I can start with bellari, which is actually Marie's favorite species, because she showed actually that species is mostly parthenogenetic. So in 90% of the case, you form diploid eggs, which actually miss the first division of meiosis. But in 10% of the case, you have a regular meiosis, which produce males. However, these males never contribute their DNA to after fertilization. It is excluded. They are only required for the activation of the egg. So we did obtain a SPO11 mutant by CRISPR for these lines, and we found that 100% now of the progeny were females and diploid, forming diploid eggs. And this can be maintained actually by fertilization with sperm from another species. Then we look at pairing. So this is the gonad of bellari in the wild-type situation. And you can see only one spot of the X chromosome and one spot of the autosome, indicating they are completely paired. And in the SPO11 mutant here, even at the end of the gonad, you can see that these 2 autosomes are not paired and the X chromosome is not paired as well. This is quantified on this scheme. You have many more chromosomes unpaired in the SPO11 mutant and on the autosome compared to the wild-type situation. And when you look at the synaptosomal complex, and we don't have all the antibodies yet. For the moment, we have antibodies only for the lateral element, which is called HTP1 in nematodes. But you can already see that you have a clear difference between the wild-type here and the mutant SPO11, where you have many more threads of HTP1, indicating that probably you do not have the central elements. And if you zoom in on this one, which are the same images but done by expansion microscopy, you can actually distinguish the 2 threads of the lateral element in the wild-type situation and only a single thread in the mutant, indicating that probably but we need to confirm that they don't have a central element. So again, bellari falls into the purple team, that what we would expect, and require double-strand break to pair its chromosome and form a synaptosomal complex. Another way of testing this correlation, where we have mutant, we just got mutant for the Oshayus, so homozygous mutant for SPO11. I just know that it's sterile for the moment, but I don't know about pairing yet. The next step was, okay, and we are going to continue that work and to do mutant in as many species as we can, sorry. But the other ways of demonstrating that that correlation would be to be able to change the numbers of chromosomes in a single species and see whether that would change the mode of reproduction. So unfortunately, to my knowledge, it's not possible to do that with animal species, but it has been done actually for the budding yeast, Cerevisiae. Actually, 2 groups managed to fuse the different chromosomes of Cerevisiae using CRISPR-Cas9, and they managed to reduce the number from 16 to 2 chromosomes, and even a single chromosome in the case of this lab, by fusing the telomeres and keeping only one centromere here. And so what we decided to do was to recover all those lines that Jeff Böke actually sent to us. And this is a collaboration with Valérie Borde at the Institut Curie. And so we obtained all these different lines of Cerevisiae, which with 2 pairs, 4 pairs, 8, 12, and 16 pairs of chromosomes. And we induce again a SPO11 mutation in each of these lines to see whether they are now dependent on the double-strand break. And the initial results are here, the following, just by looking at the viability of these spores. Spore viability in yeast is actually an indication whether the chromosomes are segregated properly. So yeast are usually haploid. You can induce them to become diploid, and then if you put them on a poor medium, they will start meiosis and form this ascus with 4 spores, which are the 4 products of meiosis. And then you can replate those spores, see whether they form colonies. And the one which have the right numbers of chromosomes will be able to form colonies. And you can see that the line with a reduced, with an engineered genome, going from 16 to 2 pairs of chromosomes, are doing meiosis pretty well, I mean, as well as a wild-type situation with a SPO11 one. The one with 2 chromosomes seems to have some difficulties. We don't know yet why exactly yet. And if you look now, the same one with the mutation in SPO11, we can see, as expected, that the one with 16 chromosomes are completely sterile. There is no spore which is viable. However, when you reduce the number of chromosomes, then you start to see the appearance of a lot of viability, actually, already at significantly when you reduce the number of chromosomes by 2. And if you have only 2 pairs of chromosomes, you see that you're almost as efficient as doing meiosis, whether or not you have SPO11. Indicating that, and you could say that by reducing the numbers of chromosomes, you also have a higher probability that chromosomes segregate fine just by chance. You have like 50% for the first chromosome, 50% for the second pair. So you would be at 25%, and we are at 62%. And for example, if you add 2 more pairs, then it's another quarter of the quarter, so you expect 5 or 6%, and we are at 50%. So they are doing it very well. And if we, and we also made a model in which we accept some extra chromosome without changing the viability of this spore. So you could expect that if you have 1 less chromosome, then you die, you're not viable. But if you could say that if you have extra chromosome, you would still be viable up to a complete diploidy. That's what we've shown here in this model. But in this case, you would expect at maximum 50% with 2 pairs, or for example, 20% with 4 pairs. And we are way above this very generous model. And as I said previously, the lower expectation would be 6%. So they are doing meiosis much better than expected. We are looking at pairing. I don't have the result yet to show how they pair, but we started to look at how they do the synaptonomal complex. And here, the central element is called GMC2, and the lateral element is called RED1. And you can see it's quite nicely that in the one with only 2 chromosomes, 2 pairs of chromosomes, they form a nice long SC, something that you never see in a yeast. Usually you have 16 tiny chromosomes, or here you have the 4 pairs also. And in the mutant situation without SPO11, if you have 16 chromosomes, you're unable to form any synaptonomal complex. You just form a polycomplex here, which is not associated with DNA. And this is the vast majority of what I saw for these ones. For the line with 4 chromosomes and 2 chromosomes are exactly the same as these ones. But in some instances, we found the formation of a bit of a synaptonomal complex. Here, for example, in these ones, you don't see polymerized threads. You see tiny bits of SC, but this is a central element, indicating that even without SPO11, they're able to make some stretches of SC. So what these preliminary results shows us is that if you reduce the number of chromosomes to 2 or 4, you seem to be able to do meiosis pretty fine without SPO11, whereas with 12 and 16 chromosomes, you're unable to do it. So the conclusions are that I think we have identified time and the numbers of chromosomes as major constraint, shaping the evolution of how chromosomes pair. And I think now these two parameters are actually linked. I mean, this is not the same challenge to pair 4 chromosomes in 5 days, for example, in a Drosophila males, compared to pairing 32 chromosomes in a few hours in yeast. This one is a bit of a bold statement so far, because it seems that 5 or 6 pairs seems to be kind of a threshold, whether you need or not SPO11, but we need more species, of course. I'm sure that additional parameters are going to be involved, the size of the genome, the ratio of heterochromatin to a chromatin, or the numbers of transposable elements, and they probably also shape how you pair your chromosome. And I also want to point out that, the major focus at the moment is, of course, on human fertility and human reproduction, but that these model organisms, such as Drosophila, yeast, etc., have really contributed enormously to our understanding of meiosis, and that has benefited the understanding of human reproduction. However, these organisms, like yeast and flies and C. elegans, have been selected as lab model because they reproduce very fast and they give lots of progeny, which that allows us to do genetics and biochemistry. However, that's not how humans reproduce, and they reproduce very slowly with very few progeny. So probably human meiosis have faced different constraints than those organisms. And I think that there is a place also to study non-model organisms with different modes of reproduction. And that could also be useful for to understand human reproduction. So I will finish by thanking the people who did the work. So Thomas Rubin did the work on Drosophila males. Catriona Munro did all the work on jellyfish, and Aurélien Perrier is doing the work on nematodes. We could not do this work without our collaborators on different species. Steffi Williams helped us to find homologs of meiosis in different species. The work on Clicia was done with Evelyn Holliston's lab. I presented the work of Eva Wenger on nematode with Marie Delattre, and the work of Johan Nicolas in Valérie Bor's team at the Institut Curie on yeast. And I will stop here and take any question. Thank you.
Hello. Very nice. I was just wondering in yeast, so in budding yeast, so when you have the situation where you have 8 chromosomes, so viability, spore viability really strongly drops. Can you try to slow down the cell cycle to see whether you would shift the limit from 6 chromosomes like to 8?
No, exactly. And it's part of the plan that we plan to do by reducing the temperature. And so in yeast, it's quite easy. If you reduce the temperature, you can double the time. But we plan to do that in a second step afterward. Yeah.
So my understanding that animals that don't require SPO11 for pairing use proteins that bind to pairing centers that are specific to particular chromosomes, so that these chromosomes can find each other by these protein marks. Now, clearly, these protein marks should not be present in yeast that have been engineered to have lower numbers of chromosomes. So how do you imagine that homologs are being recognized, even if not perfectly, at least some of the time, in your yeast with 6 chromosomes?
Yeah, in yeast, so pairing centers so far has been found only in C. elegans. We don't know if they exist in other species. And for example, Drosophila can pair its chromosome without pairing centers and without DSB as well. So that does not answer the question, but it means that those yeasts probably are able to do it as Drosophila is able to do it as well. So in Pombe, for example, something that could be conserved is they've shown that the transcription of long non-coding RNA on specific loci can be used as a glue between the chromosomes. So it's possible that you have some specific transcription site, species dependent, that could form some kind of condensate with long non-coding RNA, and that would be the glue between homolog chromosomes. But that is, I mean, it's been found only in Pombe so far. So.
We have time for one more question.
Beautiful talk, Jean-René. My question is about the first part in timing, because I always think about oogenesis as being very slow compared to spermatogenesis, just on a whole. And then thinking about that, do you think this is a developmental issue? And on top of having, as you start to think about looking at other species, does like the order of sex determination have a factor, or is it something unique about, spermatogenesis and oogenesis programs that might drive the difference as well?
Oh, you mean, so in flies, clearly, spermatogenesis is much longer. And they grow, and you can see just by cell size, they are much bigger at that stage, at least. Whether it's conserved in other species, that I don't know. In Clicia, in the jellyfish, it actually starts much later than oogenesis, and they have, but it's because they have an initial step of a proliferation before starting meiosis. But I don't know if they have started to pair their chromosome yet. I don't think so. I think it's the same in Medaca as well. In Zebrafish, I don't know, but in Medaca it's the same. They start later. Now, I don't have a general view of how, if it's conserved or not. Yeah.
Thank you very much. So our next speaker is Yaniv Elkouby, and he's going to join us via Zoom. So I'm just like looking at behind the desk whether everything's in order. Yes, I think we see Yaniv, right?
Hello.
Hello, Yaniv, can you hear us?
Can you hear me well? See my slides? Pointer?
Everyone's fine? Okay, and thank you very much for joining us. And if you would like to start.
Sure. So I would first like to thank Marie-Hélène and Elvan for the invitation and for such a fantastic meeting. And I'd like to tell you today about a story that we believe can revisit our understanding of early oogenesis. This is an entirely unpublished story that we present for the first time, so really exciting, and I'd appreciate your feedback. So my lab is taking a journey on a journey to before the beginning of life, asking how are germ cells produced. We know that developing eggs and sperm must execute dynamic but precise cellular programs for successful fertilization, but and defects in those programs are the leading cause for miscarriages, infertility, and gonadal tumors. However, the mechanistic defects are still unknown because we lack complete fundamental understanding of the natural processes. And so the overarching question in my lab is how are eggs produced? And we are focused on the earliest stages of oogenesis, which begin where germline stem cells give rise to a meiotic precursor cell of oocyte called the oogonia. These oogonia divide incompletely, leaving cytoplasmic bridges connecting sister cells, which generates a cluster of interconnected germ cells that are surrounded by somatic cells called the germline cyst. The induction of meiosis then transforms these oogonia into differentiating oocytes that will execute the first steps of meiotic prophase. The oocyte will then leave the cyst to form the primordial follicle, which will continue to grow while arrested in meiosis. Now, these are critical stages in oogenesis, since in mammals, they all occur during fetal development, and by birth determine the number and quality of oocytes for the entire person's lifespan. And in my lab, we're interested in many questions along multiple processes along this differentiation path. And to address them, we contributed to establishing the Zebrafish juvenile ovary as an excellent model for oogenesis, where ovaries are flat and transparent, which makes them ideal or really heaven for microscopy. Every little circle here is an oocyte, and all of the stages of interest are laid here for our observations. We developed methodologies for live time-lapse analysis of real-time processes within the intact gonads, for the three-dimensional analysis and of cellular and subcellular structures, as well as automated segmentation tools, again, inside the intact gonad, and also methods for isolation of stage-specific oocytes for our various biochemical and omic approaches, which altogether provides us with our holistic approach to oogenesis. Using this holistic approach, we're able to eliminate several unpredicted cellular machineries in germ cell production. The first one is a new organelle, primary coelom, that we identified in oocytes and spermatocytes, and this has been this work has been led by Avishag Maitlis in the lab, and she will tell you later on about how we were able to take this investigation from fundamental discovery all the way to clinically relevant information for human infertility. In another direction in the lab, we were able to decipher mechanisms of formation of a membraneless organelle in the oocyte. And Karin Levy in the lab was supposed to tell you today about this very exciting symmetry breaking mechanisms that lead to the formation of these organelle, and also uncover new cellular organizer in the oocyte. Unfortunately, we'll have to wait another opportunity to hear about that. Today, I'd like to tell you about mechanisms of female germline stem cells and ovarian development, a project that's been spearheaded by a super talented student in the lab, Neta Hart. And so I told you how these early stages really determine the number and quality of oocytes for the entire lifespan. And this is because, as much as we know, there is no de novo production of oocytes in mammals throughout life. And this is because germline stem cells are not preserved as during fetal development, as based on data from the mouse. We know that the flush of retinoic acid signaling and others really floods the gonad, inducing differentiation in all germ cells, leaving no maintained germline stem cells. This leads to development or synchronous development in the mouse ovary, where all oocytes are present at the same developmental stage and progress together from one stage to another, from one day to another. However, the human fetal ovary during 15 to 35 weeks post-fertilization is highly non-synchronous. You can see, we can detect meiotic stages next to presumably later meiotic stages and mitotic stages, suggesting that at least during this period of time, germline stem cells are still maintained and produce oocytes. And so we need a better model to understand these mechanisms, and this is where zebrafish enters the scene, where the ovarian development of zebrafish is highly non-synchronous. Early meiotic stages are found adjacent to mitotic stages, as well as to later developing follicles. And indeed, zebrafish oogenesis is highly conserved with that of mammals and in humans. However, there's one predominant advantage, which is the fact that zebrafish ovaries maintain germline stem cells that actively produce oocytes throughout life. However, so we can study them. However, we don't know much about those stem cells. We don't know how germline stem cells, female germline stem cells in vertebrates, especially in zebrafish, self-renew, how they produce progenitors, what is the potential niche, and we also don't know really how they produce progenitors and lineages of differentiating oocytes. An interesting answer to this question came where we identified modules in zebrafish developing ovaries where we can find consecutive stages of cyst aligned linearly within those modules, starting by a cyst of mitotic algonia stages, followed by a liptoten stage, and this is not showing the entire Z projection here, but they're here, initiating meiosis, followed by later stages, and then an oocyte that seems like it's leaving the cyst to form the primordial follicle. Now we can detect multiple of those modules in an ovary, and the whole ovary will probably take the entire wall behind me. But this really suggests a developmental linear manner where we have a young tip of the module and a old tip of the module, really predicting that germline stem cells should be found at the young tip of the modules, presumably giving rise to this module. Strikingly, we were able to locate germline stem cells marked by the universal germline stem cell marker nanos at the young tip of modules. We characterized those stem cells. We found that their size is very similar to algonia, suggesting that a subset of cells that previously were identified as algonia are actually germline stem cells. We found them either in solitary, in the minority of the cases, or in most cases next to modules, either developing modules with only algonia or more developed modules in these cases. We also found that those stem cells are, in a minority of the cases, are found as singlets, and in most cases are found in groups of 2 to 7 cells, suggesting some division dynamics or subpopulations. And indeed, with phosphohistone 3, we could find that some of them divide, as shown here, while others are not, indicating that there could be several subpopulations of those stem cells. However, we wanted to see whether those stem cells really give rise to the module. And so we used fish for various markers along the modules. We could, again, locate the nanos-positive cells at the young tip of the module, as shown here by the normalized intensity of the signal along the module. This was followed by FOXL2 mitotic algony stages, positive mitotic algony stages, which were followed by REC8 oocytes at the meiotic entry, and DMC positive oocytes in later prophase. All of those cases, the majority of each cluster was found intimately adjacent to the previous cluster, and only a minority of the cases were not, suggesting spatial linear development in the module. Moreover, multiplexing those markers really showed this consecutive development, as shown here with nanos and FOXL2 and their signals, nanos with nanos, FOXL2, and REC8 in this module, and the intensity signals, and with REC8 and DMC1. Again, quantified to show that they are adjacent, following adjacently one another, and suggesting spatial linear development. And even more interestingly, at the margins between different cell fates in the module, we could detect cells, as you could already perhaps notice, that express both markers, suggesting that perhaps they're downregulating the early marker and start to upregulate the next marker as they transition through development. Again, supporting such a spatial linear development. Now, this is all done by fish and quantified, but it's still done manually, which is prone to perhaps bias. So we took an additional approach here using deep learning assisted automated tools that Vinit in the lab has developed to analyze cysts and modules, which were previously used to analyze physical parameters of those cells. Now Vinit was able to modify those to really segment in 3 dimensions the fish signals that I showed you before. And he asked whether for each marker in close proximity in 3 dimensions, there's a cluster of another cell, and what was this cluster positive for, what marker. And we consider 15 pixels as about half diameter of a cell to indicate intimately adjacent any signal, any cluster within those 15 pixels, meaning half a diameter of the cell would mean really intimately adjacent. Anything beyond that would not. And you can see here nanos and FOXL2. The majority of FOXL2 cells are adjacent to the nanos. Some nanos are alone. Some are adjacent to REC8 signals, probably just by chance. FOXL2 and REC8 shows the same dynamics. Really, again, in a non-biased manner, conclude the spatial linear development in these modules. We next wanted to obtain more proof for this development, so we performed lineage tracing experiments where we used a flux cassette driving M-cherry combined with Hitchcock-Cree ERT. We induced recombination. We aimed for partial recombination at larval stages, aiming to induce recombination only in a subset of these VASA-positive cells. We traced those animals to juvenile stages to obtain partial labeling of those clones. Now, in this partial labeling, we wanted to see whether those modules, cells expressing consecutive markers seem to be clonally related. And it seems like this is indeed the case. Those are nanos2 and FOXL2 cells intimately adjacent in a model that are also positive for the blue M-cherry signal here. These are very tough experiments, and cherry immunofluorescence is combined with the HCR, so the signals are a bit less optimal than M-cherry alone, but still quite definitive. Furthermore, FOXL2 and REC8 adjacent cells were also labeled by the clone, which further confirms the spatial linear development within those modules. Now, we wanted to next address the morphology of this module. And I told you earlier how the progenies, the direct, or at least as far as we can tell, the most direct progenies of the germline stem cells are the mitotic algonia that construct the germline cyst. Now, this cyst is really serves as a hub for germ cell production. Many key mechanisms and processes are taking place in the cyst, chromosomal pairing and symmetry breaking all take place within the cyst. However, in contrast to Drosophila, where the construction of the cyst is really well understood, in vertebrates it is still poorly understood. Now, two major features of the cyst is the fact that the cyst clusters are surrounded by somatic cells, and that, again, based on mouse data from the mouse, cysts progress synchronously. All cells in the cyst are supposed to be in the same developmental stage. So I wanted to address whether this is indeed the case in light of the module discovery. And we found that cysts within the modules are, at least appear, very intimately adjacent to one another. To address whether those are collectively engulfed by somatic cells or separated by somatic cells, we used the somatic marker, showing that, at least morphologically, cysts of different developing stages are not separated by the somatic marker. More molecularly, we use the FOXL2 marker here and also other markers showing that FOXL2 positive cells and FOXL2 negative cells are not separated by somatic cells, suggesting that there is a more collective wrapping up of this module by somatic cells. But this is all done in confocal microscopy. We wanted to obtain higher resolution, and we generated we performed serial block phase scanning electron microscopy that generates three-dimensional views of entire cysts. Here we can see a module of early cells, leptoid and zygote cells, and I hope you can appreciate how between module cells and non-module cells, we can easily detect extra membranes and cytoplasm of somatic cells that wrap around. However, between those leptoid and zygote stages, there seems to be no additional separation. We segmented these cells as shown here, really showing the interface between the tight interface between those two cells at different stages, in contrast to the gap of somatic cells between module and non-module cells. So the module appears to be the morphogenetic unit of oocyte production, as it is collectively surrounded by somatic cells. The other feature of the cyst that I mentioned is the fact that all cysts are supposed to be at the same, all cells in the cyst are supposed to be in the same stage. However, we detected cytoplasmic bridges connecting sister cells here, and when those sister cells express different markers. Here showing FOXL2 meiotic oogonia and a cell entering meiosis connected by a bridge, as well as a cell at meiotic entry and a cell progressing through prophase that are connected by those bridges, suggesting that cysts are actually non-synchronous but are patterned, again, consistent with this module development. And so I showed you that we identify those ovarian modules where cysts are organized in a higher developmental linear organization throughout the module. The germline stem cells are found at the young tip of the module and likely give rise to the module and the developing oocytes within it. We were next interested in understanding the developmental dynamics of these modules, and so we looked at divisions that construct cysts. In these cases, we're showing cysts that are engulfed by somatic cell markers again, and you can appreciate how in some cases those cysts are homogeneous for phosphohistone 3 markers, suggesting that they're all undergoing mitosis at the same time and are synchronous. However, this was the minority of the cases. In the majority of the cases, cysts were non-synchronous. Some were positive for phosphohistone 3, while others were not. We wanted to substantiate this by time-lapse imaging, and we took advantage of a system that was developed by Amal in the lab. Amal has established a culture method, a long-term culture method for the ovary, where she could show that oocytes remain vital, as shown by these active mitochondria, through 10 days in culture. Somatic and germ cells continue to divide in culture, and importantly, oogonia cells pulse with DDU at day 1 make it all the way to primordial follicular diplotin stages during cultures, showing that this culture really recapitulates the major events of oogenesis reliably, And it now enables us to perform rapid functional screening, as I'll show you later, but also perform long-term live imaging, as we did next, where we recorded ovaries for 48 hours with 10 minutes intervals, looking for cell divisions in cysts. And we found, again, examples for two cases, either synchronized divisions or non-synchronized divisions. Following this montage, you can see how red arrows indicate no division, green arrows indicate cells that are actively going divisions, and the color-coded dots here show the daughter cells of each divisions. In this case, you can clearly see the two cells are dividing together. However, in this case, this green cell divides first, and only when this finishes to divide, the next cell begins its division. And this was actually the majority of the cases. So it seems like the meiotic divisions in the cyst that construct the modules are not synchronous. We went back to our lineage tracing experiment, where we found that clusters of oocytes that express the same developmental markers, in most of the cases, were all fully labeled by our lineage tracer. In other cases, they were all non-labeled. However, in a significant portion of the cases, those clusters showed some cells that were labeled by the blue M cherry nuclear signal, and the others were not, as shown here for FOXL2 and here for REC8. And this to us suggests that there appears to be at least two clones in each module. And this suggests that in these models, at least two clones of germline stem cells, are now showing this linear tracing with nanos as well, but at least two clones of germline stem cells somehow alternate in producing oocytes of multiple clones in these modules. Next, we're interested in perhaps the biggest question of all, which is how really this module development or development through module is spatially regulated. And so we teamed up with a group of Philipp Janker at MDC Berlin and with Hannah from his lab. We performed single-cell RNA-seq for ovaries. We're able to profile all ovarian cell types, as shown here, as well as generate oocyte developmental trajectories based on clustering and on pseudotime. And each of those stages is represented by those markers, nanos2, this is FOXL2, REC8, DMC, and a zona pellucida marker, which labels later stages. Looking for regulators, strikingly, already within the first 10 top 10 enriched genes in the nanos cluster, we identified the JAK2B ligand of the Notch signaling pathway, and Notch receptors, Notch2 and Notch3 receptors, were also enriched further below. Now, Notch2 signaling is known to regulate stem cells in many other cases. In the germline, it is known in Drosophila to regulate niche formation, and in C. elegans to regulate niche activity. So this suggests that the Notch signaling is a strong candidate to also regulate germline stem cells and oocyte production in zebrafish. And so to address this, we started by mapping the Notch signaling component in the ovaries. We could confirm the expression of JAK2B, specifically in nanos2 positive cells, and excluded from FOXL2 positive cells, as shown here. We found Notch3 receptor co-expressed with FOXL2 cells. There are also FOXL2 cells that are not expressing Notch3, and Notch3 cells that are not FOXL2 positive, so there is some dynamics here. We found Notch2 to be co-expressed in FOXL2 positive cells, and potentially some additional expression in somatic cells around. So we have ligands in the stem cells and receptors in the progenies, and as predicted, we identified the HER6 target gene of the Notch signaling expressed specifically in the FOXL2 positive cells, suggesting that they received the signal. And so this shows that the Notch pathway components are correctly positioned in time and space to be regulators of oocyte development in the module. To test this, we turned back to our culture system that I introduced earlier, which also allows us to use small molecule inhibitors to test for function. We used the DAPT Notch inhibitor that prevents gamma secretase cleavage of the receptor and blocks notch signaling. In the control cultured ovaries, we could detect nice modules with normal numbers of nanos-positive stem cells, FOXL2 cells, and REC8 cells. However, strikingly, upon notch signaling inhibition, we detected a significant increase in the number of nanos2 positive cells, a decrease in the FOXL2 and REC8 cells, and also really a near complete lack of modules being constructed, suggesting that notch signaling seems to promote differentiation and suppress germline sense maintenance in those modules. So in these modules, it seems that notch signaling suppress the germline stem cells and promotes the differentiation of progenitors, likely upon the induction of expression of the ligand in a subset of those cells, and how this is done, we still have to figure out. And so everything I showed you so far occurred during ovarian development in the juvenile fish. But I told you that germline stem cells are maintained throughout life in zebrafish, and so we wanted to see whether this development through module is also relevant for ovarian homeostasis and potentially regeneration. So we looked at adult ovaries, and strikingly, we found very nice modules in these ovaries, and as you can appreciate here, with nanos, FOXL2, and REC8, as magnified here, suggesting that during ovarian development, oocytes are very likely produced from germline stem cells through ovarian modules, during homeostasis. So this is very interesting, but it also important, since, as I mentioned before, in the mouse, all stem cells initiate differentiation and progress synchronously. However, I showed you how in humans and zebrafish, development in the ovary is very non-synchronous. And so we wondered whether these non-synchronous organization of oocytes in the human fetal ovary could also represent some order, and perhaps oocytes could develop through similar modules as in zebrafish. To test this, we teamed up with the lab of Susana Lopez and Silvia from her lab, and we could show that there seems to be such linear organization in ovarian cords, where nuclear OCT4 labels the stem cell, and then the accumulation of SYCP early in meiosis is followed, and this is in turn followed by SYCP that is already progressing or aligned along chromosome axis, indicating this progression. And we're now extending this with further markers to further establish that. And so in men, when germline stem cells are preserved and maintained, this enables reproductive regenerative medicine. However, in females, this is not the case and really provides a barrier for any such solution. So we believe that perhaps by deciphering germline stem cells regulation in zebrafish, we have a potential to identify the missing factors in humans as a first step toward female reproductive regeneration in the future. And so I showed you that we identified those ovarian modules that are conserved during development and the adult ovary. They're also conserved, seems to be conserved in developing human ovaries. They offer really a new understanding of female germline stem cells in vertebrates, uncover a new mode of oocyte production, and perhaps as promised, potentially revisit our understanding of these processes in early oogenesis. So now moving forward, we're concentrated on two major directions. One is to visualize the germline stem cells dynamic, which potentially or hopefully will uncover the asymmetric divisions, interaction with potential somatic niche cells, and so on. And in parallel, we're also performing spatial transcriptomics to the zebrafish ovary, developing ovary, together with our collaborators at the Younger Lab. And these are already real images of spatial transcriptomics in the ovary, where we hope to identify regulators with positional information along the module, which will help us to further decipher mechanisms. And with that, I'd like to thank my team. I already mentioned everyone. This project specifically was led by Neta, and she received a lot of help from Dr. Yoel Bogush, our research associate and lab manager, and my right hand in the lab. Farouk, our fish facility manager, really helps with everything we do. I'd like to thank our collaborators, our funding, and before I really finish, if I may take a moment for a few additional words, I'd like to say that, you know, I was supposed to be there with you in person, and unfortunately I could not join you because of the current circumstances. So despite that, I'd like to say that I'm grateful for the Foundation Singer-Polignac for enabling the remote talk. The foundation nobly aims to advance art and sciences, the two flagships of the human spirit. So I'm grateful not only for the opportunity to share our own findings, but also as an opportunity to showcase the same human spirit. Israeli scientists today and in the past 2 years have continued, despite everything that's happening around, to pursue their passion for science, experience the joy of science, to seek truth and generate knowledge for the benefit of the entire humankind. Every day, with every experiment we are able to conduct, with every excitement of new results, with every talk we have to deliver from a sheltered room like I am doing now, we show us to ourselves and to the world that the human spirit will always prevail. In his famous statement, Galileo said, "And yet it moves." Today I say, "And yet we scientists move forward." So I'm grateful for the Foundation's effort to help delivering this message of hope, and I wish for better days all around. Thank you for your attention. I'll be happy to take questions.
Thank you very much. And we have time for one question. So any pressing question from the audience?
Hi, can you hear me? Yes. Thanks for the talk. Is it known how the cyst actually resolves eventually in human, like how the asynchronicity ends up in then being synchronous after birth as compared to what happens in the fish?
Yes, so this is speculation, but I think it's reasonable to assume that eventually germline stem cells are not maintained anymore, and whatever progenies they were able to produce are there progressing. And even in the later ovaries in human, you know, the oocytes and the follicles are not entirely strictly synchronous at the beginning, and you can find also in the mouse a combination of primordial and primary follicles. So this probably phases out as germline stem cells are not maintained anymore. But that would be just my speculation.
Thank you very much, Jenny, for joining us, like given the circumstances. Thank you. And our next speaker is Petr Svoboda from Prague. And let's see whether things are going to go back.
So before the slides come up, I would like to thank the organizers for the invitation. I was actually so eager that I almost booked the flight last year, then realized it's in 2025. Now it will be something for completely different. I will talk about small RNAs in oocytes, and I will summarize some of the results which have been published, and I brought in some results which are new. So first of all, I will give a brief introduction to the small RNA pathways. There are three in animals. There's the microRNA pathway, where microRNAs are genome encoded, and they produce specific small RNAs with well-defined sequences, and these serve typically for gene regulatory functions. There is RNA interference, which is triggered by long double-stranded RNA, which is converted into small RNAs. And this is a pathway which typically serves for a defense. It can suppress viruses, it can suppress mobile elements, and now and then it's acquired also for gene regulation. And then there is the piRNA pathway, which is the primary defense mechanism against mobile elements in the germline, and now and then is also adopted for gene regulatory functions. The three pathways slightly differ in the molecular mechanism. Sometimes you have a dicer is the enzyme which makes small RNA, which is dedicated to the pathway. The pi RNA is dicer independent, and the small RNAs that are slightly longer. These are 22 nucleotides on average. The effector complexes, in all cases, are having a member of the Argonaute family protein. It's the AGO subfamily in microRNA and RNAi. In the PV, there's the pathway is the PV subclass. Now, in some species, these pathways adapted and merge, and they really diverge across the metazoa in many different ways. In mammals, we have a single dicer which is producing the small RNAs. But in mammals, typically the major product of dicer are microRNAs because the dicer is adapted for that, and you have this whole complex driving post-transcriptional regulation through base pairing with the targets and typically inducing translational repression and RNA destabilization. The RNA interference is relatively rare. Now, for the pi RNA pathway, in mammals, for a long time we had mouse knockouts, and all mouse knockouts of the pi RNA pathways, the females were fertile. But a couple of years ago, my and other groups made a knockout in hamsters, and it turned out that females in this case are sterile. And it seems actually that the absence of phenotype in mice might be an adaptation and very specific feature of mice, because there is RNA interference in there. There's a partial redundancy between the two pathways. And also maybe the mouse germline is not under such a pressure of mobile elements like the male germline. So we think actually that the golden hamsters may represent more the mammalian system rather than the mouse, which seems to be derived. We also had a look at the microRNA pathway, and this has been a really interesting case because it turned out that mouse oocytes don't need microRNAs, and we have been for a long time searching for the mechanism which is suppressing the microRNA function. Because what was observed was when we use these reporters, so there is a perfect complementary site to a microRNA, this target would be cleaved like an RNA interference pathway. It only requires the Argonaute with the small RNAs. And we have these typical microRNA targets with so-called bulge sites, which have imperfect complementarity. And you can see that when we have these perfect reporters, they are repressed well in growing oocytes, and we can detect the repression in fully grown oocytes, but not the reporters which actually mimic the typical microRNA targets. And so here we simply inject RNA and we monitor the luciferase activity. We see no repression. And so we have been thinking, and it's been also noticed that if you make a knockout of the microRNA pathway in the oocyte, if you knock out one of the components, there is no phenotype. So it seemed that microRNAs are suppressed or irrelevant in the oocytes. And so we were looking into this more. We were able to actually reproduce these systems with the nano-look reporters, which allowed us to use much less of the injected microRNAs, and still we would not see the repression. And we thought that there might be some repressive mechanism, and it turned out actually that the mechanism repressing the microRNA pathway is the oocyte growth. And it turned out that once we look at the numbers, we realized that in growing oocytes and fully grown oocytes, we are having approximately the same numbers of the microRNAs because the microRNAs are not adapted for the growth. Like messenger RNAs, they are having increased stability, and so they can accumulate. They actually don't accumulate. The density of messenger RNAs in the cytoplasm in the oocyte is pretty much the same as in the somatic cells, but microRNAs did not adapt, so the growth essentially dilutes the number of the 50,000 molecules, which we observe for the abundant ones, would be really well functional in somatic cells, but in the oocytes gets diluted because you have this divergence of the concentration of the targets and concentrations of the microRNAs. And I think this might be a general concept which is interesting for the oocyte field, that the oocyte growth actually put constraints on the molecular machinery in many different ways, and the systems which need to operate in the oocyte have to adapt to these constraints, to this dilution effect. Apart from that, we noticed that actually in some oocytes, so in cow and in pig, we found individual microRNAs which are having extremely high copy number. And indeed, it turned out that these are actually functional. So it seems that as long as the microRNAs reach nanomolar and higher concentrations in the cytoplasm, they might be functional. So this, for all the other species, always look at the abundancy of the microRNAs. This was one of the lessons, that there is no specific mechanism. It's simply the dilution caused by the oocyte growth and the lack of adaptation of most microRNAs to accumulate. And we still don't know actually why these microRNAs which remain there are accumulating. Now I would switch to the RNAi pathway. Mouse oocyte is actually the one of the few mammalian cell types which has truly functional RNA interference pathway. And it has been already discovered quarter a century ago, where long double-strand RNA injection into oocytes or zygotes would induce sequence-specific degradation of complementary RNAs. And it turned out that there is a unique adaptation in oocytes, because oocytes express a smaller dicer. And this is an interesting case of evolutionary adaptation, where this smaller dicer is lacking a helicase domain, one of the helicase subdomains, which make this helicase arm in the dicer highly flexible, and it actually adopts easily the long double-stranded RNA substrate, while the rigid arm of the helicase, typically in dicers in somatic cells, are not accepting this long double-stranded RNA substrate well. And evolutionarily, this happened because the long terminal repeat insertion, which provides an oocyte-specific promoter, is an interesting feature of rodents, because this family really expanded. There's a couple of 100,000 of insertions of these elements, and hundreds of genes were rewired and remodeled by these long terminal repeats. Typically, you have a retrotransposon insertion from the parental copy can go into full-length element insert, but in a lot of cases, and in this family is the majority, gets recombined, you end up with a solo long terminal repeat insert. But this is a perfect copy of a promoter, and this family is even funnier because it has a splice donor, and many of the copies have an AUG. So this is like a plug-and-play remodeling system for increasing the variety of the genes and reprogramming actually maternal and zygotic expressions, because some of these subfamilies are oocyte-specific and some of them are expressed during the zygotic genome activation. So they contribute to evolution of the maternal and the zygotic program. And so dicer is one of the many events which happen in the mouse genome. And so it happened in the common ancestor of mice and hamsters. And in the different lineages, so we look at a hamster, rat, and mouse, there were different evolutionary trajectories. So in hamster, we don't see RNAi active. In rat, we see that it is functional. In mouse, we see it's essential. So if you look at it, this is transcriptome mapping on the genome, and you can see that in the mouse there are actually two of these elements. When we delete this one, in some genetic backgrounds, this promoter is able to take over and rescue the phenotype. Again, in the rat, you see that this is a dominant dicer promoter. In hamster, we see a little bit, but the dominant dicer product is the full-length one, so we see little or almost no endo-siRNAs produced in the hamster. Now, we deleted back then the empty element from the mouse dicer. We got mimic of the dicer knockout phenotype. It's a very nice spindle defect. Transcriptome was completely misregulated. This is because RNAi in mouse oocytes was adapted to control genes, and it's actually because of the sense and antisense pairing of messenger RNAs and antisense pseudogenes, which are embedded into long non-coding RNAs. So there is hundreds actually of the antisense pseudogenes expressed in the oocyte. You see that 2/3 of all the siRNA sources are coming from this antisense pseudogene pairing with messenger RNA. Here, this orange was something what was noticed before, but was not really realized that what it is a large tandem array of the SP110 and 140 clusters. This will become important later. So if you look at the mouse, massive transcriptome dysregulation, spindle defect, and there is also some increased expression of mobile elements because there is a redundancy between RNAi and the pi-RNA pathway. Now we did the same mutant in rats. So we deleted the empty element in rats. We see much less of the transcriptome dysregulation. There is almost no overlap with the mouse. Seems all these antisense pseudogenes evolve in the mouse lineage after the rat separation. Actually, if you have antisense pseudogene and messenger RNA in two different loci, they will over evolutionary time erode. You won't form double-stranded RNA after some point because of the point mutation. So this gene regulation is very specific to mice. No spindle defect here whatsoever. We know now that it's probably kinesins and optineurin which are overexpressed in those mouse mutants which cause the spindle defect. And so some line and empty elements, so there is a shared function in suppression of mobile elements. And what was really bizarre was that out of these upregulated genes, out of top 50, 30 were tandem arrayed zinc fingers with the crab domain. And they are organized like this on chromosomes, seems to be on the tips of the assembled red chromosomes. It's 6 of them. And there are these zinc fingers which are producing somehow siRNAs. We do not figure out the source. And you see there are like one after another one. They are all upregulated in the mutant. So what we are working on right now is to test if these are functional. We can express them. We can see them in the nucleus. We don't know if they can repress gene expression. But what it seems that actually the common feature between mouse and rat, apart from suppressing mobile elements, might be control of these tandemly duplicated transcription factors. Maybe we hope that this could be a case of genomic conflict or genetic conflict where basically this is the toxin and the siRNAs are providing an antidote. So that was the RNAi. And one more thing, it's not only dicer, which is modified by mobile elements, it's also the argonaut, the effector complex of RNAi. And it seems that the dicer is modified to boost RNAi, and the argonaut, at least the best to our knowledge, may be actually providing some restriction on the RNAi activity. So around the same time as this dicer insertion caused this short dicer isoform and started to boost the RNA interference, there was an insertion of a full-length element here downstream in the AGO2 and emergence of an oocyte-specific promoter. And again, there were different trajectories in hamster, rat, and mouse. So in the rat, it was recombined out into the solo LTR. In the mouse, the full-length insertion was fragmented. And it seems from mutations we generated that it provides an enhancer for this oocyte-specific promoter, such that, and also the terminator of the transcription. Nature sometimes does weird things. So you have extremely high expression of this N-terminal fragment of the few exons, and then relatively low expression of the rest. We know that this upstream part, when we overexpress it, we cannot detect any function for it. But what is interesting is this oocyte-specific promoter and the N-terminus of the argonaut in oocytes, because there is a difference in this unstructured N-terminal tail between the somatic form, which is not expressed in the oocyte, and this maternal one. And so we were testing in some of the mutations what might be the significance of this. So because this provides a transcription terminator for splice acceptor, we took the rest of the coding sequence and we knocked it in right after the first exon. And with that, we were able, and we also knocked in the somatic variant into the oocyte-specific promoter. So first interesting thing is when we knock in the somatic N-terminus, we get improved RNAi. You see these genes go down, that's an improved RNA interference. When we boost the expression of the maternal variant, genes go up. Actually, this is less RNAi. And what's interesting is when we mutate this arginine in here, it's shown on the next one, we get again good RNA interference. These are the same genes. So we have a correlation with when we replace the maternal isoterminus for the somatic one, genes go down, they are repressed, and those are the same one when we put this single arginine mutation in there. And here you see the anti-correlation here, that there is a downregulation of the genes and upregulation there. So what's going on? We don't know. This is the model. This is the unstructured N-terminal tail. And from the alpha fold, it seems that if we use empty argonal, this terminal tail goes here into this pus domain. And from cell culture experiments, it seems that microRNA pathway is not really affected. It is perfectly complementary targets when we see weaker effect. And what it seems is that for perfect complementarity, for base pairing, the 3' end of the RNA needs to be released. That's how you get the perfect duplex. And what we think is this tail here might be interfering with the recapturing the 3' end, and it might induce maybe target-mediated decay or loss of the small RNA and reducing the efficiency of RNAi. So what we think is that one mutation basically increased RNAi potential, and this one at the same time is reducing the efficiency of RNAi. Now, these are just the preliminary data. It's supported by the mutations and it's supported by some data in the cell culture. We still wait for more data from in vitro experiments. So to sum up, so mouse oocytes are a unique model for RNA silencing pathways. There is highly active essential RNAi targeting genes and retrotransposon, and this gene targeting and spindle defect is very unique and specific to the mouse. We don't see that in rats. MicroRNAs are present, non-essential and diluted during growth, and this is probably universal principle which applies to most oocytes. And so for oocytes to maintain or get functional microRNAs, or for zygotes to get functional microRNAs, you need adaptations. An example might be Zebrafish MIR430, which is a zygotic microRNA, but that one is on a tandem array and is massively expressed from the zygotic genome activation in order to produce the required number of the copies of the molecules for the function. The piRNAs are present in mouse oocytes. They are targeting retrotransposons, but they are non-essential. We think it's because there is the redundancy between RNAi. We have some genetic data supporting that. And this might be a mouse-specific feature. I would suspect that if you look at other mammals, you most likely will find that piRNA pathways in the female germline are important and likely essential. Now, this mouse-specific RNAi pathway is a functional adaptation of Dicer, and this happened only in the Muridae family. And not each member of the family got RNAi in the end. Hamsters don't really have a functional RNAi. We see it in mice and rats. So there has been species-specific evolution of RNAi substrates and targets, and again, mouse is an exception in this case. It may be a mechanism controlling rogue gene arrays because that's one of the two common features we see between mice and rats. The tandemly duplicated transcription factor is different ones. SP110 in mice. It is the Krebsing fingers in rats. And there has been multiple contributions of retrotransposons to RNA evolution. There's the Dicer, Argonaut, both are adapted. These LTRs also produce in mice these non-coding RNAs with antisense pseudogenes. And then there are ago isoforms. It seems that mice and rats, actually I didn't say that, evolved oocyte-specific and terminus, and this counterintuitively restricts the RNAi activity. And the restriction seems to be associated with arginine residues and residue at the N-terminus. So RNAi may be essential in mice, but enough is enough, and there is actually a cap on top of it to prevent it would be too functional. So I would stop here, and I would like to acknowledge people in my group and also the alumni, because some of these projects have been going for quite a long time, and I'll be happy to take questions.
From the back.
Thank you. I'm curious about your rogue arrays that you said were zinc finger, had zinc finger domains. And I'm wondering if they're sexually dimorphic in their expression, because in zebrafish on chromosome 4, there's an array that has these kind of zinc fingers, and they are sexually dimorphic.
So a simple answer is we don't know. I don't have data on the expression of these from the testes, but they seem to be expressed from the organs only in the oocyte.
Here. Thank you very much. I thought it was quite fascinating that you find this dicer isoform that boost small RNA production, whilst the AGO2 mutation kind of restrains the function. And I was wondering, is this AGO2 germline mutation linked to the presence of the dicer O isoform, meaning that you have a looser small RNA production system but a tighter functional RNAi risk?
Linked, you mean?
Like the AGO2 germline isoform is only present where you have this dicer O.
I mean, they are both expressed only in the oocyte, so we don't see any evidence for them. So they are co-expressed. So we don't see either the short dicer isoform or the AGO isoform. We don't see them expressed anywhere else. No, so this is only in the mouse. This is only in the mouse. We see the AGO isoform, we see the same one in red, and we see similar ones appearing now and there, but we don't know if they are significant. And this short dicer isoform, we looked really a lot into the different transcriptomes, and this is really an event which occurred in the common ancestor of mice and hamsters.
Thanks, Peter. So I was wondering, do you see also that miRNA are non-essential in very large cells such as neurons?
Sorry,
the dilution effect of miRNA, is it also observed in neurons?
I don't, well, in neurons there are specific regulations, because in neurons there are the postsynaptic spines, and there are places where the microRNAs and argonauts may be aggregating. So that would be something different than we see in mice. But there are adaptations for the microRNAs. They do function in neurons.
Microphone goes there, I have a question. So like if you try to put the longer dicer isoform into mouse, that will happen. Has anyone tried that?
We put the short dicer isoform in the mouse because I thought that we will activate the antiviral RNAi in mouse. I spent about €1 million on that. We managed to engineer the mouse. So we made a mouse, we re-engineered the N-terminal part, we removed the exons, we sealed it with CRISPRs, put an HA tag. The homozygous mice die postnatally, but heterozygotes are viable. We tested 6 different viruses, including SARS-CoV-2. None of them really was repressed well. We could see in some of them that there is an siRNA appearance, but it wasn't really efficient antiviral system. We also tested the canonical RNAi, and you really have to push the system a lot to observe RNAi in somatic cells. So we would have extremely high expression of the double-stranded RNA, and we would have to reach the reads per million at a scale comparable to microRNA. So basically from 5,000 to 20,000, there we see RNA interference. Any siRNA abundancy below that wasn't really efficient to repress the target. So I think this might be a problem for the viruses. We hope that if the viruses are actually, amplifying any 10% efficiency for everyone who has a mortgage, or any percent counts, then long term. But no, we could not really get it working with the viruses well. Was only one virus we would see some effect, especially when we boost the short dicer expression even more, but nothing really spectacular. So.
I had a question here. I found very interesting this observation about the different amounts, so the importance of stoichiometry. And I was wondering if you can comment, to my knowledge, mouse oocytes are kind of the smallest in between mammals, and they're substantially smaller than many other mammals. And if you can speculate, what does this mean in terms of what you observed?
I think it's zebrafish is bigger, right? It's like really big, and I think the same applies to zebrafish. For the mammals, the bovine oocytes are somewhat bigger. Most of the microRNAs are diluted again. There is only like 1 or 2 which seem to be accumulating. We don't know why. There might be some circular RNA we failed to find, or some target on which they can bind and not degrade the target, and that might facilitate the accumulation. But we could not really figure out why there is like just 1 microRNA in pig and 1 in cow, which is going to 1.5 million copies per oocyte. Generally the dilution effect is in there for most of the microRNAs. Some have some adaptations to accumulate. We don't know why.
So thank you very much, and it's time for lunch now.